Good morning, everyone. We're going to continue the biology for grade 8, so let's get started. For today's lesson, we're going to start with exploring how plants make food. As we all know, plants are living things, so practically they feed or they make their own food. But how do they make their own food? Plants here go through a process which is called photosynthesis. What does that mean? Here the plants make food using material around them. Like us human beings, we feed on animals or plants. But for plants, they use material around them to make their food. But how? The green plants here, for an example, they use water from the environment and carbon dioxide from the air to make what exactly? To make glucose. In addition to that, they use the light which is coming from the sun in order to make their food. So the whole process of the plants using water from their environment and carbon dioxide from air to make the glucose, and in addition to that, they're using the sun as a source of light. It's called photosynthesis. So the equation here can be so simple. The carbon dioxide and water in the presence of light gives me glucose and oxygen. Okay. The plants here, we use them, or they actually use glucose for what exactly? Okay, you can tell me okay, we got the idea that plants do actually use glucose, or they go through the photosynthesis process to actually gain or to make glucose. But what do they do then? They actually use the glucose for growth and respiration. And they store the unused glucose as carbohydrate, which we call it starch. Remember the previous class when we said that human beings use glucose as a source of energy for the body, and the unused is stored in the liver and the muscles as what? As glycogen. And the other spare one is used or actually stored as fat. Here, the plants, they use the glucose for growth and respiration, and they store the unused glucose as carbohydrate, which is called starch. Okay, how do you know that a plant has been photosynthesized? How would I know that this plant went through photosynthesis? The first thing, I will grab a chemical dye or a chemical pigment. It's called iodine. So I'm going to use iodine, a chemical dye, for testing for starch, because if there's starch, that means the plant has gone through photosynthesis. The iodine here is an orange color, but it turns into a blue-black when it's added to starch. So, for an example, I took here a leaf and I added iodine to it. But before I add it, there are some steps we have to go through as an experiment. The first point here, we boil the leaf in ethanol, in alcohol. And then we rise it with warm water. What does this do? This kills the cells and removes the green color. So the whole purpose from boiling the leaf in ethanol and rise it with warm water is actually to remove the green pigment. Then we add iodine. When we add iodine, if it becomes blue-black, then that means there is starch. And if not, that means no, there is no starch. So here, this is the leaf after extracting the green color, and this is the leaf after iodine has been added. The leaves have a green pigment, which is called chlorophyll. What does this do, this pigment? It absorbs the light, and it's found in the chloroplast in the plant cells. The plant cells here, they have something. How do I differ? How do I say this is a cell, an animal cell, and this is a plant cell? Practically, you will find that plant cells contain chloroplast, which gives the green color or contains the green pigment, which is called chlorophyll, that gives the plant the green color. Imagine if human beings have the chloroplast, we'll be all greenish wandering around. Okay? This was for this lesson. Let's move to the second one, which is exploring the role of stomata. Exploring the role of stomata. Okay, what is a stomata first? What is a stomata? When you take like a leaf and you cut it and you put it under the microscope, what would you see? You would see tiny pores or holes just underneath the surface of the leaf, which allows gases to move in and out. So the stomata here, they are tiny pores or holes just beneath or underneath the surface of the leaf, which allows the gases to move in and out. Okay, when a plant actually needs carbon dioxide, so what does the stomata do? The stomata opens, allowing the gas into the leaf by the guard cells on each side of the stomata. The guard cells are acting as a gate beside the stomata. So practically, they allow the stomata, or they control the stomata, whether it opens or closes. Here are some points. The guard cells, they have chloroplast, so they can photosynthesize. When the guard cells are full of water, they open the stomata. The plants take in carbon dioxide from air, and oxygen, which is a waste product from photosynthesis, is removed through the stomata. Here, we said previously that plants take in carbon dioxide in addition to water to make a glucose and oxygen. So oxygen here is actually a waste product from the photosynthesis and is being removed through the stomata. Then the water actually escapes from the plant. The stomata here cannot stay open. Why? Because the guard cells, when they are shrink, they go back and the water escapes. Okay. Another important thing that you need to know, that the number of stomata depends on environmental factors. Like what? What does control the number of stomata? What does it, like the number depends on what exactly? On the humidity of the air and the concentration of carbon dioxide. Because the first thing the leaf does is actually intaking carbon dioxide. So it's very important to know that the concentration of carbon dioxide and the humidity of air actually is one of the things, or both things, that the number of stomata depends on. Here is a simple drawing of the stomata when it's opened. Here are the guard cells, which they are so fluffy and swallowing, filled with water. This is the cell wall, this is the vacuole, this is the nucleus, and this is the chloroplast, which contains the green pigment called chlorophyll. And this is where the guard cells shrank and closed. So this is the stomata has been closed. Now we move to this lesson. Which is looking at leaves. The leaves here have features. What does it mean? That means they have specific looking that allows them to photosynthesize. They are thin, flat, broad, which means wide, green, and have a network of veins. Those tiny, tiny lines when you grab a leaf and you actually focus or concentrate on the look of it. They all contain a pigment called chlorophyll, which absorbs the sun energy and allows or enables the plant to photosynthesize. So as long as it has chlorophyll, the leaf or the plant is able to photosynthesize. There is a method here. We use it to actually seek for chemicals in a pigment, which is called chromatography. So chromatography here is used to look at the chemicals in the pigment. Let's check the leaf structure. The first thing, have you ever noticed that when the rain falls and you have plants in your garden and you look at the leaf and you find out that, okay, even though the rain has fallen, but the tiny droplets of the rain actually slippers from the leaf and falls. It does not get observed. Why? Because even if you sense it and you rub your fingers on the leaf, you will notice that there is a waxy, waterproof cuticle, which is the outside layer of the leaf. Then we've got the transparent epidermis, this is a layer, and then we have the long, narrow palisade cells packed with chloroplast. See here, this is a chloroplast, this is a chloroplast. And then we've got the spongy cells that have a large surface area. Look over here. This is the sponge cells, and this is the large surface area. This is the long, narrow palisade cells, and this is the epidermis, and this is the out layer, the cuticle. Then we've got the lower epidermis cells, and there are large space between the spongy cells. Okay, each cell type has a special adaptations to help it perform its functional and maximize the amount of photosynthesis. For an example, the light here passes through which layer? Through the cuticle and epidermis until it reaches where this area, the palisade cells. Then the palisade cells absorb light to make the rate of the photosynthesis very high. Last but not least, the spongy cells here, they capture the remaining light. So this is a section through the leaf. Thank you all.